video, we're going to talk a little bit about how we can measure evolution within populations. For a long time, you kind of look at populations over time and say, yeah, they're changing, but there really wasn't a way to measure that until two guys worked together to develop something called the Hardy-Weinberg equilibrium and the corresponding equations. So again, just a reminder, our 5 agents of evolutionary change are mutation, selection, sexual selection, or non-random mating, genetic drift, and so on. Remember, populations are localized groups of interbreeding individuals. The gene pool is a collection of alleles within that population. So again, it's important to keep in mind what are the differences between alleles and genes, because in our equations we'll have different things that represent alleles and genes. Allele frequency is how common is that allele in the population. How many dominant or recessive alleles there are in the whole population. So evolution is changes in allele frequencies within a population over time. So what conditions would not cause allele frequencies to change? That's our kind of our null hypothesis. So if you take all the agents of evolutionary change away, then the allele frequencies should stay the same. So we have 5 assumptions for what we call the Hardy-Weinberg equilibrium. You need to have very large population size to prevent genetic drift. You have to have no migration, which prevents gene flow. No mutations. Random mating, no sexual selection, and no natural selection. Everyone has an equal chance of surviving and reproducing. So this hypothetical non-evolving population keeps allele frequencies the same, or keeps them in what we call Hardy-Weinberg equilibrium. It's our null hypothesis to measure evolution. Now, is it likely that you're going to have populations that are in Hardy-Weinberg equilibrium or going to meet all 5 assumptions? No, it's not. It's pretty rare. But it is useful as a way to measure, to see if they're changing, and then look and see which of those assumptions are not being met. So what is causing the evolution? So let's look at a trait here. Fur color in cats, black and white. Black is dominant to white. So the frequency of the dominant allele, big B, would be P. The frequency of the recessive allele would be Q. So we only got 2 alleles, so if you add the frequencies of both together, they have to add up to 1. So that's our first equation, P plus Q equals 1. If we're looking at individuals, the frequency of the homozygous dominant, 2 big P's, is P squared. Homozygous recessive, 2 little b's, is Q squared. The heterozygote, there's 2 ways we can get that. You can get big B from one parent and little b from the other, or little b from the first parent and big B from the second parent. So that's 2 times P times Q. Again, if we add P squared, Q squared, and 2PQ, we should get 1. That's our second equation. So for alleles, P plus Q equals 1. For individuals, P squared plus 2PQ plus Q squared equals 1. So if we have a population of cats, 100 cats in this population, it's a big population. We have 84 that are black and 16 are white. So how many of each genotype do we have? So you can take, because black could be big B, or big B, little b. We don't know by looking at it, but we do know if they're white, they're little b, so that's Q squared, 16 out of 100, or 0.16. We take the square root of that, that gives us the frequency for little b is 0.4. For big B, then it has to be 0.6 because P plus Q equals 0.1. So if we take our P and Q values, plug them into the second equation, homozygous recessive, P squared is 0.36, 2PQ is 0.48, and Q squared is 0.14. So the null hypothesis would be that there's not going to be any change. So if we go back and measure it, and we get these values, something is definitely going on. Something is giving individuals that are black, and particularly homozygous dominant black, an advantage. Now again here we've got the heterozygous individuals seem to be in an advantage. So sickle cell anemia is a trait. It has to do with the hemoglobin gene, the protein that carries oxygen. If you have a particular mutation, number one, the red blood cells don't carry oxygen well, and number two, they form these sickle-shaped cells. So it's a recessive disease, so as long as you have one copy of the normal one, you're going to have enough normal cells that you're going to be okay. But when your body's under low oxygen levels, like you're exercising hard, the red blood cells are going to turn into that funny sickle-shaped, and they're going to break down, and they're going to clog up blood vessels, and they're going to damage organs, and they're going to cause the symptoms of sickle cell disease, which could include death. So in Africa, actually Central Africa, we have about a 1 in 5 individuals are homozygous, or sorry, excuse me, they're heterozygous for sickle cell disease. It's pretty high. So why is the hemophilia of the sickle cell gene maintained at such a high level? Well, it turns out that there's some kind of advantage to being heterozygous. There's what we call the heterozygote advantage. It has to do with malaria. Malaria spends part of its life cycle in the red blood cells. People that are sickle cell carriers that are heterozygous are more resistant to malaria. The homozygous dominant, if you live in parts of the world where malaria is a problem, okay, then you might die from malaria. If you're homozygous recessive, you're probably going to die or have reduced reproduction from the sickle cell disease. But if you're carriers, you're less likely to die from malaria. You're not going to have problems with sickle cell disease. So there's an advantage there.